RFLP, restriction fragment length polymorphism, analysis is a molecular technique used to detect variations in DNA sequences that can affect the length of restriction fragments generated by specific enzymes. It is a powerful tool for identifying genetic variations and has been widely used in various fields, including forensics, medicine, and evolutionary biology. The RFLP analysis process involves several steps. First, DNA is extracted from a sample and purified. Next, the DNA is digested with a restriction enzyme that recognizes specific DNA sequences and cuts the DNA into fragments at these sites. The resulting fragments are then separated by size using gel electrophoresis, which separates the fragments based on their length. The gel is then stained with a dye that binds to DNA, and the fragments are visualized under UV light. The resulting pattern of bands on the gel represents the DNA fragments generated by the restriction enzyme. If there are genetic variations in the DNA sequence that affect the restriction sites, the length of the fragments generated by the enzyme will differ between individuals, resulting in different band patterns. RFLP analysis has been used for various applications, including genetic disease diagnosis, identification of infectious agents, and forensic investigations. In genetic disease diagnosis, RFLP analysis can be used to identify mutations in specific genes that are associated with inherited disorders. For example, RFLP analysis has been used to diagnose sickle cell anemia, a genetic disorder that affects the shape of red blood cells. In forensic investigations, RFLP analysis can be used to match DNA samples from crime scenes to suspects or to identify victims of disasters. The DNA samples are compared based on their band patterns, and a match between the two samples can provide strong evidence for the identification of the individual. However, RFLP analysis has some limitations. It is a time-consuming and labor-intensive technique that requires relatively large amounts of DNA. It also has limited resolution, as the size of the fragments generated by the restriction enzyme may not be precise enough to differentiate between closely related individuals. In summary, RFLP analysis is a molecular technique used to detect genetic variations in DNA sequences that affect the length of restriction fragments generated by specific enzymes. It has been widely used in various fields, including medicine, forensics, and evolutionary biology. While it has some limitations, RFLP analysis remains a valuable tool for identifying genetic variations and understanding the genetic basis of diseases.